Yeah, let's talk about properties of enzymes now. Now, we have seen that enzymes work best with cofactors, with enzymes and stuff like that. But still, there are other factors, there are other properties of enzymes, and basing on those properties of enzymes, it determines which factors should be varied, how these enzymes should be treated for you to gain that maximum catalytic activity. So number one, these enzymes, they are proteins. And like how we said, all proteins, they're affected by certain factors. For example, temperature, heat. So all proteins, they are natured at high temperature. So literally enzymes too, they're affected by temperature. Sorry. So number two, all these enzymes, they are specific in action. Specific in a way that an enzyme A that works on substrate B cannot work on substrate C. And substrates or enzyme D which work on substrate C cannot work on substrate B. So these enzymes, they are specific in action. If you want to buy Samona, then you buy Samona. I've made an advert. You should pay me. Not what works like Samona. You remember that advert. So also these enzymes, they are specific in action. Okay? And I hope by now you know why they are specific. It's because of the specific shape of the active site. The Rock and K hypothesis tries to explain that why enzymes are specific in nature. They do speed up the rate of a chemical reaction. That's why they are considered to be catalysts. If a reaction is to take place in 10 minutes, then these enzymes are going to speed it up, and within just 30 seconds you have the products. And that's how nature is, that's how life is. You eat food for you to obtain immediate energy, to obtain energy immediately. So these enzymes, they are good as far as doing that is concerned. They are effective in small amounts. It means that a work which can be performed by 1000 enzymes, 10 enzymes are enough. Should I repeat that? If you have a chunk of work, maybe you've eaten 1 kilogram of posho, and this work requires 1000 enzymes, okay, to hydrolyze the 1 kilogram of posho into glucose. Anyway, there are different series of enzymes, okay, into maltose. Okay, now even 10 enzymes can do that work perfectly well. However, now the issue here is time. The 1000 enzymes, you might get the products in just 5 minutes or just 10 minutes. But the 10 enzymes, you might get the products in 1 hour. But still, they will do catalyze them. They will do catalyze such reactions. So they are effective in small amounts. So that's, and why are they considered to be effective? It's because these enzymes, they do catalyze reactions, but they remain unchanged. The shape of the active site remains intact. As far as a catalytic activity is concerned. And so because of this, then an enzyme can catalyze reactions, you form products, and then you bind to other substrates. form products, again the same enzymes. So an enzyme can catalyze more than one reactions, but same reactants and same products. So that's why they are effective in small amounts. So they remain unchanged at the end of the chemical
Excuse
reaction.
me.
Yes.
What happens if you have excess enzymes and there is no substrate?
What happens if enzymes are excess and there is no substrate? Then the reaction rate is going to be no reaction rate if there is no substrate. But what happens if there are proteases? Then they can work on yourself, but to avoid that, then they are maintained in inactive state. But if they are active enough, or if they have been activated, then they can be denatured again and you form back the amino acids and you produce other enzymes. So that's how good it is. But if there are others, if they are not proteases, then those ones they are broken down, they are split into different amino acids, and the amino acids are absorbed, and you do carry out protein synthesis to form back the proteins. They are denatured at high temperatures, yes, because they are enzymes. If temperature increases, then these enzymes they are denatured, they lose their 3D shape, and if they lose their 3D shape, then they are rendered useless. Inactivated by inhibitor chemicals such as cyanide, heavy metals, organophosphates, those phosphate pesticides and stuff like that. They can inactivate. Work at a specific pH, either alkaline or acidic. They catalyze reactions which are reversible, and finally, their activity can be enhanced by enzyme activators and coenzymes, such as amylase enzyme. It needs chloride ions for it to be effective. So basically, those are the properties of enzymes. If there are questions, you are free to hit the inbox or you are free to unmute and ask.